Blood-forming stem cells are very rare and yet are very important clinically. There are thousands of patients whose lives are saved each year as a result of bone marrow transplants, and the active ingredient in a bone marrow transplant is blood-forming stem cells. There are thousands more patients that we could treat more effectively if we could actually grow blood-forming stem cells in culture. But no one's ever been able to do that despite decades of effort. We don't understand the environment within the bone marrow that maintains blood-forming stem cells well enough to be able to replicate it in culture. So one of the major focuses of my laboratory is to better understand that environment, with the ultimate goal of being able to replicate it in a laboratory dish so that we would be able to grow stem cells for transplantation in patients. In the past, because stem cells are so rare, we and other people had only been able to see a tiny fraction of the stem cells within the bone marrow. But a series of advances in the laboratory, including the identification of a genetic marker for the stem cells where we can actually make the stem cells glow green so that they stand out in the bone marrow, and in this case, we've managed to clear the bones in the bone marrow. So we render the bones actually see-through. You can read through the bones when we do this technique. And so that allows us, with high-end imaging and computational techniques, to image all of the cells in the bone marrow, to see where the stem cells are located, and to digitally reconstruct the bone marrow. We can actually fly through the bone marrow in videos to do close-ups on individual stem cells and to image their interaction with specific other cell types. We'd like to understand all of the factors within the bone marrow that maintain blood-forming stem cells, because when we do, it should be possible to grow those stem cells in culture. But we're really not able to grow blood-forming stem cells yet, and that's an important long-term goal of ours.